and all the participants present here and the audience. Whatever I have heard earlier is that prevention is better than the cure. So on that line, I am going to present my work here, which is just an initiative to my lab. We are working in the laboratory, we are generating a lot of the waste in the laboratory. Particularly in the line of molecular analysis, we are using a compound what we call ethidium bromide. And that ethidium bromide is considered to be carcinogenic. Rather, earlier it was used for some trypanosoma treatment, but nowadays it is not used like that. And we are generating a lot of solid waste into the laboratory. And the method for its what we call discharge is given as per the material safety data sheet, and it is that it is ultimate to incinerate it. But many of the laboratories, they don't have the incineration facilities. Also for the university too, and particularly for those universities which are at the start. So for that, into my lab, I have taken an initiative that we can manage our own waste by ourselves. So we have gone through the literature, and in the one of the literature in 2012, we could find that this ethidium bromide is going to leach down into the groundwater. Though the compost help to retain the water, but they are not going to prevent the leaching of this ethidium bromide into the groundwater. So ultimately, through the groundwater, we are going to expose to this ethidium bromide, and since it has the capacity to intercalate between the DNA molecule or the nucleotides or the, you can say, the nitrogenous bases. So they are going to create a gap between these two, and it is considered to be one of the base. And during the replication, it is going to cause some point mutation by inserting one of the nucleotide. And this is one of the basic mechanism by which many of the what we call diseases happens. So it is not a pertinent that it causes this type of the ill health, rather it can be very common. So for that, this is what the method has been given for the incineration. Other methods are also available there, which are going to retain this ethidium bromide into some matrices. But ultimately, if we discard these matrices outside, they are again going to recycle into the nature. For that, we are not going to accumulate it and then again discharge to somewhere, creating another problem. Rather, we are going to just eliminate it by degrading it. So this is the mechanism why this ethidium bromide is going to create some changes into the genetic material, which is considered to be responsible for all the features, characters of our living status. So now in this line, we have isolated the bacteria from the contaminated sites. We can see the lab waste we have kept into our laboratory, and then we are going to purify them. Then we will study that bacterium whether they are able to degrade rather to accumulate. If they are able to degrade, can we utilize this bacterium for the treatment of our solid waste or the liquid waste generated in the laboratory? So for that, this is the plan of our work. We have gone through the sample collection, then isolation, then characterization of the bacterium, identifying how many types of bacteria is there. And fortunately, we can say that though in the nature a lot of bacteria is there, but in the lab we could find only 2 types of bacteria in the waste. It means this ethidium bromide is going to inhibit the growth of the other bacteria too. So when we discard this ethidium bromide into the sink, probably they are going to also disturb the natural population of the soil around. So then what is happening? This is the media plate on which we are growing the bacterium, and ultimately we can find that only one bacterium here is there, which is not going, the growth behavior is not spreading. Here another bacterium which is forming a layer. When they are growing it, they are forming the layer. So the 2 different and distinct characters are there. On the basis of that, we have isolated, we have studied their characters, whether they have some stress enzymes or not there, So you will find that they have the catalase, which is one of the enzyme considered to handle the stress created by the toxic environment. Then we could see whether they are able to degrade the ethidium bromide or not. Ethidium bromide is a fluorescent molecule. So it fluoresces at 260 nanometer to 265 nanometer, which is the wavelength near which our DNA molecule absorb the light and flourish. So it is near to that. So just as the fluorescent property, we could see whether the organism is accumulating or whether they are growing. So wherever they are accumulating, they are increasing the what you call biomass fluorescence is increasing, and wherever they will decrease, the fluorescence around the bacterium will be decreasing. So you can see here, just this is the colored plate and this is the white, black and white plates respective to each other. So this pink color is the color of the ethidium bromide, which is going to flourish. This is the dot, whatever you look, that is the bacterial growth. So when they accumulate, they are also flourishing. But when we incubate with the time, ultimately we are finding that this fluorescence has disappeared. It means with the time they are going to accumulate as well as then they are going to degrade it. So whatever the matrix we are going to use for the treatment of this bio-waste to remove the ethidium bromide, ultimately it doesn't have the ethidium bromide, or you can say it is not having the active ethidium bromide, rather it is degraded. So it will not be acting as a carcinogen. And ultimately we could see that they are clear. So with that we have planned a just treatment flow. Waste is here and whatever the water is accumulating here, it will be delivered to this and it will have the microbial population into the matrix. They will degrade it and then after that will be discharged. And this is the filter which will prevent the microorganism to flow into the discharge. And to apply this, we have to go for the biosafety analysis, whether this bacterium is pathogenic or not. Unfortunately, we got that this bacterium is non-pathogenic, but I cannot
your time
reveal
is
the
over.
name.